So I'm going to talk about a slightly different set of crops than Gerard has talked about. And I'm going to hark back to my most favourite crops to work on, the legumes. Gerard has already introduced beans here, but am I going the wrong direction? Okay. He's talked about beans. First of all, let me say that about 300 million tonnes of legumes, food and feed legumes, are produced every year around the world. That's dominated by soybean, the oilseed legumes. But peanut comes in pretty quickly there as well. There's a fairly large amount of peanut produced as well. Dry beans, Gerard just talked about those, largely in Brazil. And the crops that I'm going to spend a bit of time talking about, peas, which are produced largely in Europe, but they're produced in Australia as well, I might say. But chickpea, which is produced largely in India. Cowpea, largely in West Africa, mostly in Nigeria, but in some of the surrounding countries as well. And those are the three that I'm going to talk about in my short time that I've got here. But I just wanted to put those food and feed legumes in context. Peas are a feed legume largely, whereas chickpea and cowpea are mostly food legumes used by human populations. Now, the three peas, pea itself, chickpea and cowpea, which I'm going to abbreviate as pea for the rest of my talk, they're all high protein content for human and livestock, so they're one of the reasons that I work on them. They have low glycemic index starch. Those three account for about 25 million tonnes of product every year. And they have other field benefits as well, like fixing nitrogen and being very important in crop rotation. And even the stubble that's left over after the grain is harvested is high value for livestock. But there is a but in all of this, and that is yields are abysmal. Usually 0.3 of a tonne to 0.5 of a tonne. If you get a tonne per hectare, you're very lucky. Whereas something like soybean, the average yield of soybean around the world is almost 3 tonnes per hectare. And something like cowpea is 0.3, a tenth of that. So the options that are available to farmers who store these grains, and I'm really very interested in the stored grain pests for these crops, is that the options that farmers have is not to control the insects that damage these in storage and rely on the seed's natural resistance. That's a very high risk strategy because most of these legumes have very little inbuilt resistance to insects. Their second, and that's why the amount of grain that's available for human consumption can be very, very low after insects have damaged them in the field and during storage. They can use insecticides. Many countries, and then the countries that I work in, can't afford the insecticides anyway, and many people don't want to use insecticides. So then there is the possibility of using gene technology to introduce inbuilt resistance into these to protect them against insects, and that's what I'm going to talk about. This is a typical grain store. These are granaries, or places where the grain is stored in Burkina Faso, a country that produces cowpeas, as do other adjoining countries, Mali, Niger and Nigeria. And all countries store their grain in these kinds of granaries. And weevils get into these stores very, very easily, as you might imagine. So the objectives then of my research over the last several years has been to make insect-resistant gene cassettes for pea transformation, whether it's chickpea, pea, or cowpea. Transfer these insect resistance genes into the peas to confer resistance against the pests, of course, and then do molecular analyses, which would include biosafety analysis, and then field trials to confirm levels of resistance and introgress the transgene into new varieties via conventional breeding. So I would supply a line to the breeders in these countries, and then they would introduce the gene from my line into their own best varieties for dissemination to farmers in those countries. We started out with a pea weevil resistant pea. This is Pisum sativum. And the rationale behind this work was that pea weevils develop in the pea seed. Starch is the weevil's source of energy. They digest that starch with the enzyme alpha amylase. And our predicted strategy was that we should genetically engineer peas with an amylase inhibitor gene from beans. So that was the starting concept. You can see the sort of damage that pea weevils do to pea seeds there. And it's very difficult to sell these seeds, even for stock feed. And certainly Australia can't export peas if there's a single damaged seed in a shipment, for instance, when the peas are exported to largely the subcontinent. We use agrobacterium to transfer the genes into peas. A typical construct is shown here. A binary vector, shown here with left and right borders, a selectable marker gene, and then the alpha amylase inhibitor gene, which has been remade for expression in the seeds, in the cotyledons of the pea, because the insects live in the cotyledons, in the seed only. A typical transformation system, way of getting new genes into one of these legumes, is for us to start with dry seed, which we imbibe overnight, shown there in panel B. The seeds are split in half and exposed to agrobacterium, and then transferred into media every 2 weeks for about 6 months until we get plants, pieces of tissue that start looking like plants, eventually do look more like plants over here on the right hand side, and they develop roots. Getting this. They develop roots in tissue culture. We move them into little pots like this with high humidity, and eventually move them out into the glass house where they develop flowers and pollens. That typically takes us about 6 months to go from this top position here to having plants like this, to get the gene in the first instance. It then takes us a further 4 to 6 months to get this fixed in a Mendelian fashion and stabilized. So these are some of our first results that we obtained with peas. This is a picture of the offending insect here, Lupus bizorum. And here are some of the first seeds that we obtained, shown down here. This is a blot, a Western blot, where we use antibodies made against the amylase inhibitor protein. And you can see that the seeds that have the protein do not have any damage. And the seeds that don't have the protein of the gene they will harbor insects. Now this protein is made in the peas, and it is restricted to the cotyledon. It's not found anywhere else in the plant. It's not found in the stems, leaves, flowers, pollen, pods, or seed coats, but it is found in the cotyledons of the seed. So that's a function of the gene promoter that we use, that it is restricted to the seeds. It accumulates in the seeds in the expected sort of fashion, in the same way that it accumulates in bean seeds. These are days after flowering. 10 days after flowering, the protein is not detectable in the seeds, but by 14 days it's starting to accumulate, and it's fully accumulated in the seeds by the day 35 that's days after flowering here. When we then germinate those seeds, it disappears in the way in which it disappears from bean seeds. We're monitoring here days after germination, 0, 7, 14, 21, 28, 35 days after germination. This is the disappearance of total protein in the seeds, because protein is moved out of the seeds into the germinating seedling. So the nitrogen that's stored in the seed is recycled into protein in the developing seedling. So this protein behaves just like it does in peas, just like it does in beans. So these are some results from one of the first field trials that we did with these peas in a city called Wagga Wagga in Australia. It was done about 10 years ago. And this is the difference. This was an extreme year in which there was very bad insect damage, which is shown on the right-hand side there in the non-transgenic parent line, and the transgenic parent line is shown, the transgenic line is shown there on the left-hand side, a very big difference. We've moved the same gene, exactly the same gene, into chickpeas, because chickpeas are also subject to weevil damage. In this case, it's a different genus and species. This is Calosebruchus chinensis, also called the Azuki bean weevil. The non-transgenic is the insects emerge from those seeds, but from the transgenic, there's two transgenic lines shown there. The insects emerge, or very few insects emerge from those seeds. And the same, we transfer the same gene into cowpeas as well for another insect pest called cowpea weevil, which is Calosebruchus maculatus, a relative of chinensis, the one I just showed you for chickpea. And the way in which Larry Murdoch, my colleague at Purdue University, assayed the seeds of these plants when we produced them was to measure the days to emerge as adults, shown there. In the non-transgenic parent line of this cowpea, it takes the insects 27 days to emerge as adults. So every month there is a new cycle of insects developing. There is a landrace which the breeders have tried to use in their breeding programs. It's a non-transgenic PPU. It's not a commercial variety, but it does have some resistance, but they have never been able to use it because, as you can see there isn't very much difference. It only takes 38 days for them to emerge there, in that case. But the transgenic lines, most of them, they stop the experiment after 120 days because by that stage all of the insects are dead. There is one line there where the insects do emerge after 55 days. But this correlates very closely with the level of protein that's expressed in this particular line. So every transgenic event has a different amount of protein. This one had a level that's a bit too low, whereas the other ones all have sufficient expression of the transgene to give full protection against these weevils. Now this protein that we're using to protect these different kinds of peas against these different storage pests is quite different to the proteins that Gerard just largely talked about. Because proteins like the BT protein that has been used in maize and cotton and other crops, and the Roundup Ready protein, they're all proteins that are soluble in the cell. They don't go through the endomembrane system. They don't go through the endoplasmic reticulum, the Golgi apparatus, etc. Whereas this protein, this alpha-amylase inhibitor protein does. And it gets glycosylated, hence the title sugar. There is sugar coating on this protein, whereas those other proteins do not have sugars attached to them. This one does have sugars. And the first sugar that is attached to them is this quite complex looking one here, this M9. The sugar is attached to this asparagine, or abbreviated to N-linked glycosylation in plants. These 9 mannoses, and then N-acetylglucosamines, 2 N-acetylglucosamines are attached to this asparagine. And then as the protein moves through the endomembrane system, some of these mannoses are removed. Goes from M9 to M7, 2 are removed up here, to M6, to M5, and then further removals of mannoses, and then further additions of other sugars, such as xylose. In this case, this xylose is attached to this mannose here, and fucose. So it is quite complicated, quite complex. There's a huge amount of sugar is added to the protein and then gradually removed as the protein moves through the endomembrane system in the plant. This addition and subtraction of the sugars is probably involved in the stability of the protein within the cell. So when we looked at the sugars, we looked at the sugars on these proteins after we'd made them using mass spectrometry, which is shown here. And I'm not going to go into this in a great detail, but to just show you that this is molecular weight of one of the chains of the alpha-amylase inhibitor. And the size of these peaks here gives you a feeling for the relative amount of the different kinds of sugars that are attached to that protein backbone. So this is the typical pattern of the protein from the tender green bean. This is the variety of bean that we originally got the gene from, tender green. Now if we look at the alpha amylase inhibitor that's produced in the pea, and these results were the things that first alerted us to the possibility that there might be some problem with this protein after we'd made it, is that the profile looks different, especially in the ratios. For instance, there are extra peaks here compared to the bean, for instance. Some of these peaks are much higher than they are in the bean, and there are several additional ones in here. So this alerted us to the fact that these proteins, the protein that we were making in the pea, did look different to that was made in the bean originally. If we look at this, the same thing for the cowpea, for instance, I haven't included the chickpea one here, but it's still the same story. Again, the alpha amylase inhibitor that's made in the cowpea is again different to the original bean, and different to the pea. So it's starting to appear that there is a specific sugar signature, if you like, for each legume. So I'm just identifying what some of these are here. So this particular peak here are molecules that have no sugar attached to them. It has all been removed from the molecules. And then some of these other peaks here, and I'm not going to go through all of these with you, but just to summarize that some of these are 2 mannoses with a xylose attached, 2 mannoses with a xylose and a fucose, 7 mannoses, some molecules with only 7 mannoses, some with 6, some with 5, and various other combinations. So we can account for all of these peaks and identify them as what they are. So when we summarize some of these results, I've just summarized them for the transgenics, but then this made us go back and look at, well, what is the sugar signature for this protein in other beans other than tender green? And to our surprise, and it shouldn't probably have been a surprise, but it was a bit of a surprise to me anyway, that they all have their own distinctive signature as well. So here are 4 non-transgenic beans, fairly common variety, and many of you would know. Tender green is the one that we isolated the gene from, but cannellini is commonly used, being very popular in Italy. Red kidney, very popular around the world, and pinto, also pretty popular. But they all have their own characteristic sugar signature. They also have other modifications that I'm not going to spend time going through. But just to summarize then, what we've observed is that if you compare, for instance, first of all, tender green bean, the original source of the gene, and the transgenic pea, just one of the different peas that we're talking about, and then look at these post-translational modifications, these removals basically of sugars, you find that in tender green bean, 11% of the molecules have no sugars still attached. But whereas pea, the transgenic pea, has 30% of the molecules without any glycan. So this is different. However, if you go to pinto, it has more than tender green has. Looking at one sugar attached to the molecules, 56% of the bean proteins have one sugar. Very similar in pea, but a little less. But cannellini has less still. 2 glycans. 33% of the molecules in bean have 2 glycans, but pea has less, 17%. But on the other hand, red kidney has even less still, at 0. And these C-terminal truncations, again, bean has only 1% of the molecules with truncations, whereas pea has 37%, much higher level. But cannellini has 52%, higher still again. So I think you can see what I'm getting at here is that the kind of modifications that we saw in the transgenics, you also see in other non-transgenic varieties of beans. Now, Marsh and colleagues in the UK analysed the peas as well independently of us. We sent them the pea material, but they analysed the glycans or the sugars on those peas using very different methods to the ones that we used in Australia. They used enzymatic methods to remove the sugars, didn't use biophysical methods that we had used. So it's an interesting comparison to see what results they come up with independently of us. And they started off by just measuring the sugars on the major seed protein in beans. This is shown here. This is phaseolin, which is the major seed protein called vislin in peas. But they looked at the sugar residues attached to phaseolin in two different bean species here. I'll just show you what those are. Lima bean and peppery bean. They're different phaseolin species, not just varieties, but different species. But again, you can see that they have their own characteristic profile here. Again, for instance, if you look at a peak like this here with 6 mannoses, the level here in peppery bean is quite high, very low in lima bean, and so on as you go across all of the various sugars. They looked at the tender grain bean and pea as well, and basically confirmed the results that we had, that the fact the profiles were different. Again, as you can see, something like this one here, fairly minor in the pea, pretty major here in the bean. So that's pea and bean. This is the purified alpha-amylase inhibitor from transgenic pea and the non-transgenic tender grain bean. Then they subjected their data to multivariate statistical analysis and came up with results as are shown here, where most of the variations accounted for by these two here. Here's the lima bean, here's the peppery, and here's the transgenic pea, and here's the tender grain bean, each having their own characteristic profile. They did the same thing with 3 different phaseolus species and 3 bean varieties. Again, they have the lima bean and the peppery bean, tender grain, and then 2 British varieties of beans called masterpiece and blue leg. Again, showing that they all have their own characteristic profile. So this work was done independently of the work that I just talked about that we did in Australia. So our conclusions from the work that we've done on these beans is that the bean gene for protection against stored grain pests is effective in 3 transgenic pea types. So we've done the test with cowpea weevil, pea weevil, and adzuki bean weevil. The proteins, they have protein signatures that we face, of course, biosafety and regulation, and that's why this project, GMSAFOOD, was such an important one, looking at the biosafety of these, doing further tests, kinds of tests that we couldn't do to show whether these were safe or not. But if they were safe, if they do prove to be safe by all the tests that are going on, then we would propose to conduct field trials to test for efficacy and seed storage, particularly of the chickpea and the cowpea, neither of which have been tested in the field yet. All of the tests we have done are in vitro tests, even with insect bioassays. Then we would consider other things such as insect resistance management, and by that I mean the very likely possibilities that insects will build up resistance to these genes, these proteins. So we need to have a second mechanism of action, a different mechanism of action. So that would be a challenge for us for the future. But if these would work, all of these would work, then we could consider gene integration and seed dissemination, themselves major challenges. There are excellent plant breeders in the countries that I work in India and in West Africa. So gene integration will not be a major issue. But seed dissemination and stewardship, which Gerard mentioned as well in passing, that it would be very important for these, if these were to be released as well. So I'd like to finish off by acknowledging GMSAFOOD again for the work that they've done in this area, and to acknowledge the funders of this work, the African Agricultural Technology Foundation, USAID, the Rockefeller Foundation, and of course GMSAFOOD for the financial support, and the Network for the Genetic Improvement of Cowpea for Africa. This is an in-kind group who contribute a lot to this as well. And I'd like to acknowledge my immediate colleagues, Canberra Food and Feed Legume team in CSIRO. And thank you very much for your attention.
Thank you very much. And there are questions? Yeah, good. Please.
Ralph Lampson from Braunschweig. Thanks, TJ, for your very convincing talk. Would you suggest that every transgenic plant should undergo a metabolic profiling, at least to a limited extent? And that would be sufficient to rule out any health implications?
No, well, would metabolic profiling satisfy all questions? Well, it is a very hard question to answer, to know where do you stop? What is the limit of metabolic profiling? These days, the technology that's available in genomics, proteomics, metabolomics is enormous. The question then is, how much of those tests should you apply? Well, I think it will depend on the gene, the transgene that you're using. I think we need to make some rational decisions about those as to whether or not they're likely to prove dangerous or not. So I think you need to make a sensible assessment as to what it is you're going to do. And it'll vary, I think, for every gene product. In our case, this was an obvious one to do, to look at the sugar profiling, because this protein is very different to all the proteins that have been approved up till now, because it has the sugar residues on there. So I feel that it was important for us to do that kind of profiling. We published this back in 2005, and it created a great deal of angst, for me especially, but for lots of other people as well. But I wouldn't have it any other way. I'm glad we did it. It has delayed everything by many years, but I think that's what we should be doing. It's part of the scientific process.
Thanks.
Yeah, there.
Thanks, TJ. What is the effect of the maturity of the grain at time of harvest, or sampling, preparation, on the profile of the glycans or the glycan complexity? Is there a lot of noise in your data sometimes? Or?
So he's asking the question about, does maturity stage have much effect on the sugar levels on these proteins? And there can be. We do a lot of our work on single seeds to look at it. And there's very little difference between single seed glycan profiles and the mixed population. The Shuri document looked into that in great detail, even more than we did, but both sets of data agreed with one another at that level as well. You do see some changes if you harvest the grain very early. Say the season is very short, and this happens in Australia, when they run out of water and the seeds are mature prematurely. They're smaller in that case, and you do see a difference in profile then. But it's pretty rare. If they go through to physiological maturity, which is what we want to do normally, then the profiles are very simple, identical basically, but just variety specific.
So, TJ, I think you gave a very enormous amount of data between you and Peter Shuri's group that really provided information most of us haven't had before about the variance in asparagine-linked glycans on foods that we commonly eat. And those foods really don't have negative health implications. And I think that when you look at the great diversity and you see that we're eating all of those varieties of beans and peas in different geographies, different genetic backgrounds of human beings, if there is such a thing. I don't know, there's been a lot of mixing over time. But it should give us some assurance of safety, I think, in terms of looking at that kind of difference. So if you would not have provided that data, and you just would have looked at a minor difference between the tender green alpha amylase glycan pattern and that of either the pea or the cowpea, probably there would have been a lot of questions. And so I think it is important that you did the survey that you did and Peter did, and hopefully many of us will take great comfort in that.
There is a, well, I don't want to take credit for us doing more than many other people do. There are lots of published papers in the literature now showing that in fact transgenic plants have probably less variation than conventionally bred plants if you start doing the metabolic profiling that Jörg is just referring to. For instance, there's a huge literature, particularly in wheat, but in many crops, maize, some very lovely work from South Africa on this, that in fact there's greater variation amongst the different varieties produced by conventional breeding than there is by genetic engineering. But the regulations are very different for the two kinds of crops. And it's gradually, I think, getting into people's consciousness that there is tremendous variation out there already. And that GM may in fact be less variable than conventional breeding.
Okay.
Perhaps Austria from Germany. Congratulations, this is very nice result, the results here present. But I have a question. Do you have an idea about the digestibility, the availability of the amino acids? Or did you make some studies in this field? I think monosaccharides or carbohydrates and amino acids is always a problem.
Yes.
Do you have some results in this?
So yes, so the question has to do with, are these proteins digestible to animals and to ourselves? And I haven't done these studies myself because they've been done by other people. There is a lot of data, for instance, on the amylase inhibitor protein itself, as to how digestible it is. The very nicest work probably has been done by a Japanese group, published in Japanese, unfortunately. But the tables you can easily read, you can easily see the data there from the table. But the protein, if you look at it from the point of view of an allergen, potential allergen, and this is probably what you're getting at, how digestible is it in the stomach with pepsin, this protein is particularly stable. It is not easily digestible by pepsin. So that is one of the reasons why we were particularly interested in looking at the sugar composition as well. I don't know what role the sugars play in the digestibility, but it was one of the reasons why we went ahead and did those measurements. I suspect that the protein is fully digestible further downstream, and that people do get the full benefit of the amino acid further downstream, but it's in the upstream stage that they're still relatively stable, and therefore a possibility, possible allergens. And I think this will come up again more and more in the subsequent discussions when we talk about potential allergenicity.
Yes, then Jean?
Yeah. I was just wondering about the approach you took. So you mentioned earlier about the fact that this was endomembrane and that this was different processing and post-translational modifications an issue. Do you think that people are going to continue making new GMs based on this technology, or are they considering using different processes that won't have to, they won't have to worry about these kind of modifications?
But the question is, should you have to worry about them anyway?
Okay, that's a good one.
And
Maybe
well,
not.
Maybe not. But I think that already a lot of transgenic products are in the pipeline that do in fact have sugar attachments now. Many of them are for therapeutic purposes. They are being produced for use, for instance, monoclonal antibodies. Other antibodies are being produced for use in therapeutic uses. And they are all glycosylated. All of those proteins as they are naturally. So I suspect that there will be many more of these will be tested. And in fact, so this is the stuff that we've been doing on the amylase inhibitor just happens to be one of the first ones, but there are many more in the pipeline. So I think this will, the data that we have will have some influence on that too. But I don't think it's, my own feeling now, based on the data that I'm hearing, is that it's not going to be a major problem. But it is something that needed to be looked at. And I'd be interested in your opinion on this.
Well, I think it's an interesting approach to take, where you get a very high concentration in a very specific compartment. And for me, what's interesting is that this may not be an issue, may not be a problem because of what you've just shown and others are showing. But I'm just wondering if we may run into problems that we're not thinking about now, that has to do with other modifications, other post-translational modifications, and maybe we could see that. And I just wonder, I mean, the reason I'm asking the question is to question whether it would be better to approach the system in a different way and make a GM where you know that you're not going to get these differences. So you don't have to consider them, you're not going to change any of the structures, natural structures that you're trying to develop.
But if you're making, if I was making, say, a vaccine that absolutely needed the post-translational modification for AIDS, for instance, and it was going to work, if I knew it was going to work, then I would be tempted to take it through if it was going to be of great benefit and from everything that we could see, it posed no risk. So I'm not so sure that I should not do anything other than make soluble proteins. Some of the soluble proteins may be much more dangerous than a glycosylated protein. Some toxins are soluble proteins, have no post-translational modifications to speak of, yet are intrinsically very toxic. Plants produce lots of them to protect themselves from herbivory of every kind. Everything's out there to get them, to eat them.
Do you think that there's a way, I'm just thinking in terms of what you're doing when you insert the gene, do you think that there's a way to control post-translational modifications? Is there something that you can insert alongside that really controls, you can control where you put the gene, for example, or how many genes that you insert? But could you control this? Because your point's valid. What if you really need it for a vaccine?
Yeah.
Example.
But yes, in this particular case, I could remove those amino acids that get glycosylated. It is known, the epitope, if you like to call it that, the 3 amino acids that are essential for a carbohydrate to be attached to the protein are known, and I could modify those and remove the carbohydrate from them altogether. I haven't done that, but from other stuff that I know, I predict that protein then wouldn't survive in the cell. The protein does play a role on the stability of the protein. Sorry, the sugars play a role in stabilizing the protein in the cell. Certainly true for many mammalian proteins as well. So.
Okay, there are 2 questions here. Is this just a comment or?
Well, it was partly to go into that, but maybe the other question first, but I would like to come back to the issue.
Then you're next. Eva, have you a comment?
It was very interesting your talk, and I know from the literature that there are 2 types of amylase inhibitors, the type 1st and type 2nd for amylase inhibitor, and they are different in the function. For instance, the type 1, as I remember, specific to the insect amylase, and the type 2 is specific to the mammalian amylase. And do you know something about the glycosylation pattern of these 2 types? Because they are in the function, how they can inhibit the amylase of the mammals or amylase of the insects are different.
Yes, you're quite right. It's that there are multiple alpha amylase inhibitor families. There's in fact 4 of them. But we have also tested amylase inhibitor 2 as a possible candidate because it doesn't bind to human amylases, whereas amylase inhibitor 1 does bind to it. So I was very interested in the possibility of using amylase inhibitor 2. However, Murphy's Law applied, and amylase inhibitor 2 did not give any protection against the P. weevil. So there is specificity there, even within the insect groupings as well. I haven't tested amylase inhibitor 3 or 4.
Okay.
If I could come back to the glycosylation question, because there are good questions coming. I think it's important to remember that what Dr. Higgins and others showed was that the same kinds of asparagine-linked glycans are present in all three of the kinds of transgenic peas that they generated, as are present in other beans and peas that we eat. And as far as we know, that's not leading to any deleterious effect in the consumer, as long as you cook the foods. I mean, remember that peas and beans mostly have to be cooked quite thoroughly to inactivate some of the protease and amylase inhibitors and things like that, or you do end up with some problems in the mammalian gut. I think it is important to think about the context. These proteins are consumed, and our exposures through the mammalian gut, where you take care of you digest a lot of proteins that may take getting into the intestinal tract before it's digested. It may not absorb very well in the intact form. If you're talking about a therapeutic protein that's going to be injected, or maybe through nanoparticles would be absorbed as a drug, then you really do have to be very careful of those specific glycan structure. Because if you have a non-human glycan structure on a protein, such as erythropoietin, or some other protein that's important for your human function, and you put a plant glycoprotein on, you could well induce an immune response or some other kind of effect. So in those cases, it's a very different product. It's an injectable, or it's meant to be absorbed and have effects on the human. And that's not exactly the same as the dietary protein. So there is a place to really dive deep into those things for safety.
Okay, thank you. Other questions, comments? Yeah, there's one there.
As the only question I have is, you mentioned the acreage and the acreage of chickpea and peas and all compared to soybean. So actually, as industry, we would never pick up such a project like focusing on a small scale crop, since you will never get the revenues back to justify the development costs that you would put in. And my simple question is there any idea about putting such approaches in commercial products, or is it more or less meanwhile an academic exercise?
It's definitely not an academic exercise as far as I'm concerned. I am very keen to release these through the public system, if possible. If they are found to be safe, if they pass all the regulatory issues, that's my aim in life. I'm at that age where I'm worried about the time taken to go through this regulatory process, but I feel that it is important to do it, of course. But the reason that I'm shown this slide in this format was because I think that there are a few instances where countries might undertake to do this. You make the very important point that no company is going to do this because they're not going to make any money from these kind of crops. These are definitely in the orphan category. Certainly everything below peanut would be an orphan crop, I think, if you think about it. But I picked out the dry beans, which Gerard mentioned. It's very important in Brazil. And Brazil has done exactly as I was predicting here, that they might undertake that as a country. It's very important to them. It's very important in several other countries too, of course, but Brazil particularly. And I picked out chickpea in India because it is #1 producer. The Indian government has a policy now that they want to feed their own population, and chickpea is their most important source of plant protein. Probably the most important source for the 700 million who can't afford meat. The 300 who can afford meat probably don't eat very many chickpeas. And the same for Nigeria. Cowpea feeds 200 million people in sub-Saharan Africa as a protein source. And again, Nigeria is the major producer, and the Nigerian government is showing interest in the possibility that they would do that through the public system in that country as well. So it's still very risky, it's still very difficult to do because of, as you said before, the cost of regulation. If those same costs apply for these crops in these countries, then it's going to be very difficult. But not enough to make me give up yet.
Yes, I have one question around this glycosylation, whether the changing or the different types of protein or with the different glycosylations, whether the insects react differently to them. Are they as efficient as inhibitors or amylase?
Ah, yes, this is a good point. Well, when we measure the activity of the amylase inhibitors just in vitro using amylase itself, we see no difference in the different profile. The only other answer I can give you, because I don't know how else you would do this, was we have tested, of course, the insects on the seeds.
Right.
And they seem to be as effective as we predict they should be. But that's all I can say. How do you talk to the insect and say, this isn't toxic as you, would you prefer if this was less toxic? I don't know how.
Well, it would be to have them isolated and you have the insect amylase.
Oh, do the insect? Yes, I've done that with the pea amylase, with the pea weevil, not with the other two. But I can do that with the other two as well, and there is no difference.
Okay,
Yeah,
so the
so it
glycosylation
doesn't
doesn't affect the inhibitory capacity.
No. Just
Okay.
this-